going to move into genetics, which is an absolute, perfect dovetail into this and the genetic work that we've been doing. And this is going to be a bit of a round table and handoff. We have three people who are working on this. I'd like to introduce them, including Eric Caldera, who is speaking to us from UCLA and took the lead on our genetics chapter. Eric is, Eric, you are a National Science Foundation postdoc researcher in Paul Barber's lab, another dear friend at UCLA. John, hey John, good to see you. John is talking to us all the way from Hawaii. John took over from Steve Carl.
All right, so as Turney mentioned, what I wanted to do today was just talk about this really nice genetics chapter that we put together. And our aim here was to basically highlight the various genetic tools that are available for Mola research and what kind of insights we can gain from that. So what I wanted to do is just start off with introducing a few terms here. So starting with the mitochondrial genome. So all animals have basically two genomes within them. Within our mitochondrial organelles, we have a specific circular genome because the organelles themselves evolved from bacteria. And so this the mitochondria has a genome that isn't a ring like a bacteria would, and that's in contrast to the nuclear genome, which is the larger chromosomal genome. Within the mitochondrial genomes, we have something that we call the D loop, which is basically the origin of replication of the genome, and this is really useful because it's sort of prone to slippage and varying. So over time, that gives us an amount of genetic variation that's really good for revolving species level taxonomic issues. So, in the past, some of the ways these tools have really helped us is we can really solidify, the history of the three different Mola species. And separate them out from Ranzania and Masturus, and it's a really nice, clear phylogenetic result, both across the mitochondrial genome, but then also within just this D loop, we can sequence just a small fragment of that D loop and get this really nice species level classification. Also, when it comes to Pcr and Gel Electrophoresis, so this is just the process by which we are amplifying DNA, and it usually precedes any sequencing of genomes or fragments. But one of the nice things about, again, this D loop region is that it really does separate out species nicely. So, for example, in the Mola, alexandrini A and B clades, you can almost separate them out just by looking at the fragment length of the gel electrophoresis PCR. So moving on from the mitochondrial work, we recently have a fully sequenced genome. We only have one, but for the Mola. And that's really helpful because moving forward, it means that we can do smaller, lower pass genomes using the initial reference genome. And then also I would say one thing that unfortunately hasn't been used quite that much is microsatellite DNA. But there is a nice paper describing microsatellites and molas. And so basically, we've talked about these mitochondrial and D-loop sequences. They're really good for, taxonomic species level identification. But these microsatellites really can tell us a lot more about population level genetic diversity and gene flow. So I wanted to just highlight, you know, one of the kind of questions we wanted to tackle using these really robust D-loop sequences. And as Dr. Sawai has talked about, he's done some great work on Mola morphology. And one thing that we found sort of interesting as we were writing up this chapter is that we came across this species, or sorry, a population of molas in Ceuta, Spain, that seemed to just be kind of an outlier in terms of what we think of as the typical kind of morphology. So in Mola alexandrini, we typically associate with this kind of bump head feature and this bump chin feature. And then in the Mola, we have it kind of lacking the bump head and the bump chin. But in this Spanish population, we kind of had a hybrid sort of morphology where we see the bump head, no bump chin, and then we're also kind of seeing this wavy clavus that we typically associate with Mola. So to sort of resolve, you know, where is this kind of bump head individual sitting? We reconstructed the phylogeny using these D-loop sequences, and we get a nice clean result, you know, showing how robust these sequences are, where we get the, you know, this sort of bump head Spanish population falling out really nicely with the Mola clade, definitely not falling out with alexandrini. So a really clear result there. And the other thing we did while we were working up this chapter is just really do as much sequencing of this D-loop on the samples that we currently had. And we got some interesting results out of there. For example, Ranzania seems to have a degree of geographic separation from the Sargasso Sea versus Hawaii, although not absolute separation. We also found some interesting results, some, you know, larval samples that we, you know, expected to be like these alexandrinis in Ecuador, you know, one turned out to be Masturus. And so moving forward, we're sort of expanding on this, adding additional samples to get a global sense of what's going on. So I'll kind of end this section with, you know, what we can do and tools available to us moving forward. So I would suggest that, you know, given that we have a nice, robust nuclear genome for Mola, like I mentioned before, we can answer a lot of questions by resequencing lower pass genomes. One area that we haven't quite entered into in Mola research is doing transcriptomics. So basically genetics where we're looking at genetic expression that's happening, but you know, it's a stepwise process. You know, we first need you know, the genomes to be sequenced before we can get into transcriptomes. And then finally, I wanted to say just a bit about eDNA. And eDNA is the notion that you can just sample from the environment, sequence DNA, and get a sense of the species that are present. And this is becoming more and more popular in marine systems because it's you know, basically a non-invasive way to get monitoring data. And then what I wanted to add finally with this eDNA, your work is only going to be as good as the database underlying your eDNA. So historically, there have been some problems with the databases not exactly having the right Mola species names and things like that. But I'm happy to report that we did recently, you know, sequence a barcode the popular MyFish primer for each one of these clades, and that paper is in press, and I'll be happy to share that. So moving forward, it'll be good to have accurate species barcoded, and those should be in GenBank shortly as well. So at this point, I'm going to hand it over to John, if that's possible.
Thanks, Eric. All right. Everybody, I'm Jonathan Whitney. I am a geneticist and pelagic ecologist at NOAA's Pacific Islands Fisheries Science Center in Honolulu, Hawaii. And you got the sun coming up behind me. It's getting a little brighter. And today I want to jump in and share with you kind of what we've been learning about the geographic patterns of the different Molidae species based on all of the phylogenetic work that has been done, including some new results presented in this chapter. So we're going to start with the Mola, the three species of Mola, starting with Mola alexandrini. So the geographic ranges of these the species, particularly the three Mola species, has been very tricky because the two species alexandrini and tecta have only been described in 2018. So most of the specimens that are in collections are labeled Mola, even though some of them are not. So in order to kind of start looking at what we know from genetically confirmed IDs, we put together these maps which show the point locations for genetically confirmed individuals of occurrences, the size relative to their sample size. And then these polygons, the colored bits, the red here, is kind of our working distributional ranges based on both genetic and morphological data. So starting with alexandrini, we see that they have a really wide distribution in both north and southern hemispheres, temperate and tropical, in all three basins. And starting in the Atlantic, we have records as high up as southern Norway and Sweden, into the Mediterranean, down around South Africa, and even into Oman, and seem to be pretty widespread across the southern Australian, West Pacific, and then up north into Japan. But what's going on in the Eastern Pacific is kind of less clear. So our recent analysis that Eric just mentioned confirms that alexandrini is in the Galapagos and coastal Ecuador, but we don't have any confirmed IDs in the Northern Hemisphere in the Eastern Tropical Pacific. So currently unclear if they exist there. And then looking at kind of genetic partitions within the species, it's kind of a mixed bag. So Yukiko Yoshita's work showed that there was some cryptic divergence in between the Northeast Japan and the subtropical Taiwan population. But Mayan and Igard's work looking across the coast in Australia and New Zealand didn't detect any structure down there, so which is a much broader geographic area. So we have some indications of within basin divergence and also some contrary evidence. And really the more global analysis that Eric just presented based on that phylogenetic study really shows that there's no clear geographic structuring across pretty much the majority of the planet. So we find individuals from Spain mixing, clustering with individuals from the Galapagos, which would suggest that there's just one big, large panmixtic population with very little gene flow across the ocean basins. But, I think the lack of samples here, many places we only have one or two samples, really highlights that we need more samples, and we need to keep working on this to see if there's any more refined structure. Okay, so moving on to Mola. So this in the literature is what was originally described as Mola clade B in a bunch of genetic papers. So they are globally distributed in both temperate and tropical seas, but primarily in temperate. And with samples, genetically confirmed samples going as high as central Norway, down into the North Sea, all across the Mediterranean, South Africa. And then Miana's work has showed that Mola is not actually very rare, or is pretty rare in Australia and New Zealand, where the other two species are much more common. And then it is currently the only ID that we have from Western Atlantic and Florida is the Mola. So it's the only one that we've confirmed that occurs there thus far. And then this geographic structuring in ocean basins has now been a pretty robust result from a bunch of different genetic studies that have all detected this structuring of two populations, one in the Atlantic of Mola and one in the North Pacific. Very little divergence within this North Pacific population. So this is kind of the Kurashiro population. And then we get this Atlantic population that does not have much distinction within that group, and may include these individuals from Western Australia, but there's not enough samples to really confirm that yet. So we do have two distinct populations of mola across the globe, in Atlantic and Pacific, essentially. So moving on to tecta. I'm losing my mouse here. Tecta has a much narrower distribution, as Mayana was highlighting earlier. So they really appear to be Indo-Pacific, with this core distribution in the southern hemisphere, going from South Africa into Australia, New Zealand, and then recently our work confirmed that they're in the coast of South America, including Peru, and very likely Chile. So predominantly southern hemisphere, but as Mayana just told us, these records in the northern hemisphere are increasing and very encouraging. So there's only been the single genetically confirmed specimen in California so far, but we expect that several more genetically confirmed. So it's still unclear how established Mola tecta is in the northern hemisphere. It's still really cool to see how this plays out. Okay, looking at the three Mola species together, we see that all three species overlap in South Africa and Australia, New Zealand. And interestingly, with the two Mola alexandrini and mola, what's going on in Eastern Tropical Pacific is still a big question mark. Currently, all the records for mola are only in the northern. We don't have any records in the southern hemisphere and the Eastern Pacific. And then vice versa with alexandrini. We only have records of South America and none in North America or Western Atlantic. So they seem globally distributed, but based on what we're seeing so far, have not been confirmed in these sort of large swaths of the Pacific. But it's certainly, this could be data limited because we just have so few samples, and even, yeah. So we really need more samples and further genetic studies to really resolve what's going on with their geographic distributions. But the splitting of the multiple species in mola kind of has made all of us very curious to see if similar things are going on with Ranzania and Masturus. So with Ranzania, they are still a single cosmopolitan species in temperate tropical seas in all three ocean basins. And really, there has not been a thorough phylogeographic survey of this species yet. But we do have some kind of preliminary insights from what we've run on the genetics so far. For example, Mayana's recent work compared Hawaii samples to Spain and South Africa and basically found no significant difference between these populations. But this is a pretty small sample size of, what was this? This is, 12 individuals looking at CO1. And as Eric had mentioned, we did just recently detect significant difference between the Hawaii population and the Sargasso Sea, which is encouraging. And looking at, this is not genetic data, but back Fraser Berner in the 50s did propose that there are potentially two subspecies of Ranzania, a sort of Pacific population and an Atlantic population, which could include that Sargasso Sea. So there are some morphological differences among basins that would further support this kind of cricket species in Ranzania. But really, it's hard to say. We need a thorough global phylogeographic survey to really show that. But we're working on that and continuing to gather samples, which are really valuable. And then last, Masturus, also still considered a single circumtropical species, but we haven't done a comprehensive phylogeographic survey of these guys either. But we do get a little bit of insight from the few studies that have been done that really use Masturus as an outgroup for the mola. One of those both found with very few individuals. This is a sample size of 5 individuals in both of these studies of bats, and then Mayana's work showing no difference between a sample in the Western Atlantic and Florida and the Japanese population. And then similarly within the West Pacific, Mayana's recent paper found that there was no genetic structure among about 50 individuals in Australia region with the Japanese population. So no obvious structuring among or within ocean basins with Masturus so far, but these are really preliminary accounts with in most cases only a few samples. Now, Mayana's work is providing a lot more samples in Australia and New Zealand, so we're gaining ground in many places. And then all of that really just highlights that we've learned a lot from the genetics so far, particularly with the phylogeography and the phylogenetics, but we're still very data limited and sample limited. So please keep the samples coming, and we're incredibly grateful for the countless people that have contributed specimens that we are, grateful and would love to get more all the time. Okay, I will pass it back off to Eric.
Oh, and I
Okay.
think we're also, hi, Eric.
So I think at this point we were going to kind of transition.
Oh, I think we also have Mayana who's going to be adding in. Yeah?
Yes. I just have a few comments here, and that is, of course, that it's so important for us to get a global view of the sunfish species distributions, and genetics can really help us. And one of the problems that I have run up against with my work was that I sampled like crazy in Australia, thinking I was dealing with one species and I could do population genetics. And of course, I ended up with 4 species and not enough of any in any one area to really do anything proper. So I have this idea of an ocean sunfish consortium where we share samples, basically. And so I pitched this idea to all of you researchers out there. And what it is, it's basically a global collaboration sharing samples for genetic analysis. And I thought it could have 2 phases. Phase 1, where we just do a central database of samples and see how that goes. And if that's successful, perhaps in future, we could look at doing a Molidae biobank, basically physical storage of samples. And the reason why is like most researchers don't have enough samples by themselves at this stage to answer a lot of these questions that are coming up. And the quality of a study often depends on the numbers. So if we pool our samples, we get better science. And we can also make use of these individual samples that are sitting around the world. And we can streamline the metadata in this database. And we can, most importantly, I think, have pre-agreed guidelines for collaboration. And also, the way that I thought it could work was that you maintain control over your own samples. So being part of this doesn't mean you relinquish control of your samples. And in future, of course, if we go to a Molidae biobank, it will allow us to basically safekeep a lot of samples, avoid loss, have consistency in storage, and also centralized place where the public can send in samples. How will we do this? Well, I think that it's important that such a collaboration would build on clear guidelines and trust, and then really focus on the advantage of collaborating. And it needs to be incentive-driven for everybody who's part of this, and it needs to be fair and clear with a win-win for everybody. And I thought such a consortium could meet regularly and brainstorm and pitch ideas to studies and funding. And then if somebody has a funding idea or a study idea, could pitch it to the rest of the consortium and say, does anyone else want to join my study? I have this amount of samples and I can see you guys have those. Who wants to join? And you can then, as a participant, as a consortium member, choose whether you want to participate or not in each individual study. And so, of course, if you join and have enough samples, there's a co-authorship in there. If you only have 1 or 2 samples, you could get a consortium authorship and an acknowledgement, basically. So that's how I thought it could work. And so how to become a member would be simply sending in information to this database, and this would be available to all consortium members. And the database, I thought, could just consist in the beginning of curated metadata, stuff like, has the sample been genetically ID'd? Do we have the morphology of the fish? Is the specimen available? Is there a photo available? What kind of fixative treatments have this sample undergone? And which other studies has it already been used in, with references to GeneBank and BLD and so forth? So this is my idea of how we might start a global collaboration. And if you are interested, please register your interest by just emailing me. My email address is here. And to go forward, if there is enough interest, I will take the lead and develop some draft guidelines and a format and organize an online meeting where we can brainstorm. And then people can put up their hand if they want to be the database manager or have some other role. And then we can really knuckle down and develop the final guidelines and get MOUs, memorandum of understandings written up and so forth. And then just get in amongst it. So that's all from me. Tierney, come back.
Hi. Well, that's great. That's great. Thank you, Mayana. I think it's really, it would just be great to collaborate and to put all these things together in one place, and with universal collection standards, protocols, all that. So I do champion these efforts. I love this idea, and I hope we will make that email available on YouTube. And whoever registered, we will. Here it is. Thank you, Joe. Right there. Please do write in your interest in starting this biobank, because I think it would just be so useful. Because often, like what happened with our genetic samples, Steve Carl passed away, sadly, and we moved to John Whitney. John, you've been ensconced with all sorts of additional work obligations in Hawaii. So then we moved the samples. I hand carried them to UCLA, and now Eric's taking. So samples do switch hands, and it'd be great to have one place to put them all. But I just wanted to thank all of you for jumping in for the genetics chapter, because it's that's certainly an ongoing area of research, and so many more discoveries to be made. Yeah, I think we have Eric I just wanted to let people know Eric is a little bit delayed because he's having a little bit of technical issues. But let's see. We did have some questions from the audience. I did want to do a shout out to some of the students who've been tuning in, Sarah, and tuning in from, I believe, Denmark. Then we have Leo and Mimi and Helen and Nicole from Colombia. So thank you guys all for tuning in. Definitely keep your questions coming through YouTube. We'll do what we can to answer them. Someone did ask where they should send their genetic samples, and I would say, write to that email that we had up there. You can also write to Eric and myself. I'm just tierneyteese@gmail.com, and we will make sure that those genetic samples get put in the big pool that we have right now. So, okay. Well, thank you guys all for tuning in. Eric and Jonathan, we'll sign off to you guys.